So thanks for organizing this panel discussion later on, and thanks for having this chance to talk here. So today I'm going to present an alternative approach to come up with counterfactual placebo, that is to use this Recency test. So this is joint work with Deborah and Jim, who are from the Fred Hutch, and also David Glendon, who is from UCSF. So the main goal of this talk today is try to construct counterfactual placebo instance for active arm trial based on Recency test. So here we would like to emphasize this main talk is focusing on active arm trial. That is, we're considering only the trial with one arm, that is the active intervention we are interested to evaluate efficacy with. So later on in the discussion, we'll briefly discuss how to extend it to the more commonly applied active control trial where there's additional active control arm in the trial. So to come up with a counterfactual placebo instance, our proposal is to apply this HIV recency test to all those HIV positive subjects we observe in the screening population. In that case, based on a number of estimators I will introduce later, then we will be able to estimate a pretrial instance. And our proposal is to use this pretrial instance as a counterfactual placebo instance for those trial population. So in this talk, I'll first introduce how we can estimate instance based on this Recency test. Also, I'm going to derive the assumption and identifiability of this proposed instance estimation. Under what circumstance will they to serve as counterfactual placebo instance for the trial population. And finally, I will show a brief summary of our sample size calculation results, and also to show some examples of in the future we would like to design some trials if this proposal will come up with reasonable sample size to achieve power to analyze certain hypotheses. So I will start with this instance estimate. So this instance estimation with Recency test, or is also called this cross-sectional instance estimate, has been widely applied in estimating disease instance from cross-sectional surveys. So namely, we will almost collect all samples as a cross-sectional time, so approximately at the same time. And then for those disease positive, we'll apply some, we'll obtain the biomarker value of them. And since those biomarkers will be related to recent infection, therefore this recency test will be giving us some information on whether the subjects will be recently infected or not. Particularly, here are some notations. Suppose that as a cross-sectional sample, we'll collect a total of N subjects. And in the setting of HIV, suppose that we know N plus to be HIV positive and N minus to be HIV negative. And suppose that's based on this recency test, we're able to identify total NR number of recently infected subjects to be HIV recently infected. In that case, a cross-sectional instance estimator is given by this formula. So it is quite simple. So in the numerator, we have this observed number of total recently infected subjects. And in the denominators, we have two terms. One is those HIV negative subjects, and the other is a parameter related to the recency assay property, which is called this mean window period. It is the mean duration of recency test positive for HIV infected subjects. So one intuitive way to understand this estimator is that we are dividing the total number of recently infected events versus their total person year of spending in that recency period. Therefore, we obtain an estimate of instance by this estimator. So one restriction of using this estimator is that we will assume all long infected subjects will have zero probability of being identified as recent. Otherwise, this omega will not be finite. However, in practice, not a lot of recency assay will have that property. And what people do is they come up with a modified estimator to address for this false recency type of issue. That is, there are some positive possibility that long infected subjects will be identified as recent. So particularly, we may let T to denote a cutoff time to determine whether a subject is recently infected or not. So basically, if a subject has been infected less than T years ago, then the subject is truly recently infected. While on the other side, if it's infected longer than T times ago, then it is falsely, it is regarded as non-infected or not recent. And in that case, our definition of mean window periods will reduce to this mean duration of rinse infection. So basically, this serves as an estimate of this mean duration of rinse test positive for those truly positive subjects. So for those subjects who truly infected less than 2 years ago. And this beta T, which is called this false recency rate or FRR, is the probability of rinse test positive for randomly chosen long-infected subjects who's infected more than 2 years ago. When we look at, after some analyzing of this estimator, we find out this estimator actually is an unbiased estimate of this weighted instance in between time negative T to this current time. So it serves an estimate of what's going on this cross-sectional sample. So a number of assays has been proposed for this indicate HIV recency, and then they can be used to estimate HIV incidence from sample collected in cross-sectional samples. Particularly, there are some assays, for example, LSEIA, the BET assay, or the lag avidity assay. And more recently, researchers start to combine different assays to form the so-called multi-assay algorithms, or MAA. Therefore, this MAA would have better performance in identifying whether a subject is recently infected or not. So for example, in our later exercise, we'll use this recommended assay. That is, a subject will be identified as recently infected if they have lag-ability assay smaller than 1.5 ODN and very low greater than 1,000 copies per ml. So based on this knowledge on making use recency assay to estimate an instance, we can then come up with a design for active arm trial. So basically, our intuition is try to make use of those HIV positive subjects we may see in the screening population to come up with this. Particularly, so suppose that in the screening population, we have HIV positive subjects and HIV negative subjects. Intuitively, for those HIV negative subjects, we will enroll at least a proportion of them to this active arm trial, and then we will follow them up for a period of time such that we observe an incidence. So this lambda 1 hat will be indicative of this the trial population receiving this active PrEP intervention. And we call it this PrEP incidence lambda 1. So in that case, what this is like really simple, this lambda 1 hat will be served as an unbiased estimate of this lambda 1. On the other hand, we are trying to infer this counterfactual placebo incidence lambda 0 using this recency test. So particularly, we will apply a recency test to those HIV positive subjects. And based on the estimator I have shown before, we may come up with this lambda 0 hat as an estimate. Then a natural question would be, under what assumption can we link this lambda 0 hat with the estimate of interest? That is the counterfactual placebo instance lambda 0, which is for the trial population who will be included in active arm trial, their instance if they actually receive no PrEP. So next, I'll spend some time trying to link this lambda 0 hat with lambda 0. And one strategy is that we will look what other estimate we can formulate for this lambda 0 and how, what kind of assumption do we additionally impose by making use of this lambda 0 hat instead of other estimators. So to estimate this placebo instance, the gold standard will be to use a concurrent randomized control. So this is may not be ethical in this current HIV prevention setting, but it'll be a gold standard for our exercise to see what can we obtain from this data and what other settings what other settings are requiring compared to this setting. Here we'll let time 0 to be the screening time or the randomization time for a trial. And suppose that at time 0, we will recruit those HIV negative subjects who will be eligible for the trial and who are consent to take this PrEP intervention in the trial. We will randomize them at time 0 and then to 2 arms. One is this PrEP arm, where the subjects will receive the active intervention, and we can have an estimate of lambda 1 hat. And for the other group, they will receive no PrEP, and the observed incidence in this arm will serve an unbiased estimate or the gold standard for estimating lambda 0. So however, as I've said before, this trial may not be ethical or may not be feasible to conduct. So our next example will be a crossover trial. So this crossover trial will be very closely related to this registrational cohort idea Sheila has presented maybe 1 or 2 weeks ago on this seminar series. Suppose that at time -t, we will enroll a number of HIV negative subjects. And then we ask them to come back at time 0. And then we are trying to ascertain their HIV status at time 0. So we know the HIV status of all the subjects at time 0, and for those HIV negative subjects, we'll recruit them to the trial and then follow them up for this PrEP intervention. So one idea to construct this counterfactual placebo instance is that we may estimate it by the observed HIV instance over -t to 0 among those who return at time 0 and eligible and consenting to the PrEP intervention at time 0. So comparing to the concurrent randomized trial, this crossover trial, the estimate from crossover trial may subject to some potential bias. The first one is here, obviously, we're comparing 2 time periods. So if there is structural time trends over time period, then probably this 2 time period may not be comparable. So we'll see some bias from there. And another issue is that here we are in a closed cohort setting. In that, for example, for those HIV negative subjects we recruited at time -t, if their HIV risk, they are highly variable. In that case, those who are with higher HIV risk will be more likely to experience event during this early period. Therefore, if we may come across this so-called selection bias, in that the subjects we recruited in the trial at time 0 will be more likely to have a lower HIV risk compared to those subjects we evaluate this no PrEP instance over time -t to 0. So other than these 2 sources of bias, there may be other settings where we need to be careful in conducting this crossover trial. For example, people, they may have some tests from time -t to 0. So they may be test positive before time 0 and may not return at time 0 before because they know their status. And also they may initiate PrEP in -t to 0. So in that case, we're not estimating the no PrEP incidence using the data from -t to 0. So those are 2 trial designs we may consider. Now we get to our main proposal, that's to use recency test. So first we start with like a simple setting where suppose that we have a perfect recency test. That is, even though we're based on the information we collect at time 0, whether subject is recently infected or not, we have perfect information of whether these subjects will be infected before time -t or after time -t. So that is no misclassification in recency assessment, such that we can accurately determine that. So in that case, there is some linkage of this perfect recency type of data and also the data collected from crossover trial, in that we're trying to ascertain the subjects who are infected from -t to 0. The only difference is that from the crossover trial, we're collecting data prospectively. So basically, we first look at from among all those who are HIV negative at time -t, and then we follow them prospectively. But for this perfect recency test, we are looking at retrospectively, in that we are tracking all those subjects who come to the screening at time 0, and we track whether they are infected before time -t or not. So in that case, we're not restricting this population to be those who are HIV negative and -t, and we are actually looking at the open cohort, in that this cohort will be refreshing by the population. Therefore, by using this perfect recency test, it may help to alleviate part of this selection effect type of issue. So other than that, this perfect recency test type of setting have maybe subject to a similar set of potential bias as in the crossover trial. So that is, we would be careful about the structure time trends. We would there may be some trouble if people actually test positive in between time -t to 0. Therefore, this screen population may not capture what's going on in this population, in this target population. And also, we if people initiate PrEP, we are still not estimating a no PrEP incidence there. So finally, we'll look at a more practical setting where in practice, there's no perfect recency test. We cannot really ascertain whether a subject is infected before or after time t. In practice, there may be false positive and false negative recency test results. So in that case, there are subjects who are actually recently infected, but based on that recency test categorized as long infected, and there's the other way around. So in this setting, if we let P to be a constant HIV prevalence, lambda 0 to be a constant HIV incidence of interest, and also suppose that lambda 0 is small so that we can approximate this probability by lambda 0 times T. So in that case, if we write out this probability corresponding to different observed category, so for example, here we have 3 different categories observed at the time 0. So for example, for those HIV negative subjects, we are observe n minus of them, and the approximate probability will be 1 minus P, just 1 minus the prevalence. And for those HIV who are identified as HIV recently infected by this Recency assay, that the total number will be NR. So in that case, its probability will be associated with the prevalence, with the instance lambda 0, and also related to omega T and beta T, which are related to this property of this Recency assay that's related to false positive and false negative probabilities. So if we make use of this approximate probabilities, and by maximum likelihood estimation, we actually find out the MLE for lambda 0 is given by this. So which exactly the HIV incidence estimator proposed in Hutchinson et al. 2012. So this is the estimator we're going to use. So we are showing that if we have a constant prevalence and incidence assumption, and also if we're able to make some approximation, then this estimator will be estimating lambda 0 we would like. So to summarize, so here I have a slide trying to summarize what kind of assumptions we're making to use this counterfactual instance based on recency assay. So first of all, it's a big chunk of assumption. That is, we assuming the distribution of HIV risk exposure remains the same in the group of period assessed for this instance, cross-sectional instance versus the group and period receive active product. So for this big assumption, we will need, for example, there's no structured time trends. We will need the constant instance component prevalence in the period of time we're interested in. And also, for example, we will need this willingness to enter trial screening is independent of HIV status. So we don't want to have the setting where a subject, after they know they're HIV positive, they won't come to screening. Therefore, it will mess up with this instance estimation based on this cross-sectional sample. So also, we would like to have no PrEP used in the screening population. So this is obvious since we want a no PrEP instance estimate. And finally, one added assumption is that we really need accurate determination of MDRI and FR. So those are two key parameters that will take into come into play in constructing this instance estimator based on recency assay. So there are really a lot of people working on trying to determine accurate property parameters, MDR and FR, for the recency assays. And now people are trying to determine them in different subtypes. All right, so now let's come to our next part. So I have introduced this instance estimator, and also under what kind of assumption would this instance estimator unbiasedly estimate this counterfactual placebo instance. Now we're going to look at more closely what the study design will look like. So this is, in principle, very similar to the graph I had shown before, but now I have put in more notation here for the aid of computation. So suppose that for this trial design, we would like to screen in total n big N subjects. And this big N will be the number, will be the sample size of interest, since it will decide how much people we need to screen in total and how much resource we need in order to conduct this trial. And suppose that based on the prevalence of HIV, there are maybe N plus HIV positive subjects and N minus HIV negative subjects. So for this HIV negative subjects, there are a certain proportion, our proportion of them, we will enroll them to the trial. and we may follow them for up to for 12 years of time, and we may observe in total n events. And this is just a simple estimator of the instance we observe in the trial, that is to divide the total number of events versus the total person years in the trial. Well, on the other part, for the counterfactual based on recency test, we are going to apply recency test to those HIV positive subjects, and then we are going to observe NR as the number of recency subjects. And we're going to estimate this counterfactual placebo instance by this lambda zero hat, this estimator I have shown before. And the only difference is that we're going to replace this omega t and beta t by their estimators. And those values are available from literature, their mean value as long as their associated variabilities. So based on this trial design, for analysis, a natural approach is we will try to contrast lambda 1 hat and lambda 0 hat to come up with a measure of efficacy. So here we are trying to do the same thing. It's like we are trying to look at the efficacy that is defined by risk ratio, R equals to R1 over R0. And this quantity can be estimated by R hat, which is just taking the ratio of these two estimators we obtained from the last slide. So we are looking at a hypothesis testing, R equals to R0 versus R not equals to R0. So this R0 may be 1, indicating that null hypothesis we're interested in will be like this intervention will be not effective at all. But here we propose to have a smaller R0. For example, in our later example, we use R0 equals to 0.5, which means that even in the null hypothesis, we are considering 50% effective. The main reason is that in this current setting of HIV prevention, we are looking at more effective prevention measures. Therefore, it may not be satisfactory to have an intervention which we know is better than no placebo at all, but rather we want to have an intervention which is highly effective. For example, it may be 85% effective, and we want to rule out the probability that it'll be even 50% effective. So the test statistic will look like this. So basically, we're taking the log estimated risk ratio minus log R0 and standardized by its estimated standard deviation. The calculation of sample size is quite standard. We just try to revert test statistic to come up with the alpha level beta power test corresponding to this alternative hypothesis identified by R equals R1. So to show some results, first we consider, suppose we're trying to propose a future trial for the MSM, Manhattan sex with men, transgender women. And this may be one possible combination of population of interest. And this is very similar to the trial in PTN-003. So basically, we're collecting the cohort is composed of US, South Africa sites, and also Asia sites. And those different regions are associated with the different subtypes of HIV. And for recency tests, usually it performs differently in different subtypes. So for example, for this test algorithm, their MDRI and FRR are quite different in different subtypes. So based on all those information, we're then able to come up with the key parameters we'll need to calculate the screening sample size. The overall incidence and prevalence in this population is 4.4% and 50%. And the overall MDRI is 141 days, and the FRR is 1.0%. We'll assume that 85% of HIV negative subjects will be enrolled in the trial to receive active treatment. And the hypothesis we're looking at is R equals 0.5 versus R equals 1.15. So 50% effective versus 85% effective. So we calculate sample size for 0.05 alpha level and 90% power, and this is the screening sample size look like. So if we follow all subjects for 1 year, then in order to make use of this recency test constructed counterfactual placebo instance, we will screen a little less than 2,000 number of subjects. And in that case, if it's under alternative hypothesis that is this intervention is 85% effective, we will be expecting to see around 29 Recency test positive subjects and 9 events in the active arm trial. Well, if we would like to elong this follow-up time to 2 years, then we will screen a little less than 460, a little less than 1,500 subjects, and we will see approximately 22 number of events in HIV Recency test positive events and 14 trial events. So those numbers are, so for example, for this total screening around less than 2,000 subjects, that will be a sample size that's reasonable and easy to handle in designing this trial. So in another example, where we consider a young woman trial in sub-Saharan Africa, and for this area, it's dominated by subtype C, and it has slightly lower incidence, 3.5%, and slightly higher prevalence, 25%. So in this population, the total screening sample size is still manageable. It's less than 4,000 subjects need to be screened to conduct this trial. To summarize, so we have, in this project, we have derived sufficient condition for estimating counterfactual incidence in active arm trial based on Recency tests and proposed methods for calculating sample sizes. So our results suggest that future one-arm trials with counterfactual placebo incidence based on Recency assay can be conducted with reasonable total screening sample sizes. So particularly here, I want to highlight the assumption we need for estimating this counterfactual placebo incidence using Recency assay. So first of all, we will need no prep use in the screening population such that the estimator can be indicative of a placebo. And the other assumption basically we're making is that this distribution of HIV risk exposure remains the same in the group and period assessed for cross-sectional instance and the group and period receiving active products. So one thing I would like to remark on this is that this proposal of making use Recency test to the screening population will make a huge change in this screening procedure. So traditionally, screening is just for examining eligibility criterion, and most importantly, we only care about this HIV negative subject who will be recruited in the trial. But here, this proposal is quite novel in that we not only care HIV negative subjects, but we also care those HIV positive subjects in the screening population. Therefore, this existing screening procedure may need to be adapted a lot, trying to capture, unbiasedly capture those HIV positive subjects in the screening population. Finally, here in this talk, I'm focusing on this active arm trial, so only one arm in the trial. But more commonly, we would like to consider active control trial. So we'd like to include additional active control arm in the trial. So the inclusion can be because of the safety reason. We want to compare and see if this active intervention have any safety issue. So in that case, we can still apply this type of design and just adjust this sample size calculation. And the only primary endpoint is still efficacy based on this counterfactual placebo instance. However, if based on this active control trial, we're still able to observe the instance of HIV under this active control intervention. So, and there may be some other way to try to combine information from that arm into this design as well. That is, how do we combine two efficacy as defined by a counterfactual placebo instance versus the instance we observe in this active control arm. So that will be all for my talk today. Thank you so much for listening. So our paper has been submitted as well, and you can also see it at arXiv, and I have linked the website here. Thank you.